Astrophysicists wonder about the enormous number of bodies out there in the universe. And here on planet Earth, we biologists wonder about the enormous diversity and numbers of species of living things, animals, plants, and microbes. There are millions of species. So many, in fact, that we don't know how many species there are, which is truly remarkable for the year 2025. How do they all evolve? Why did they evolve in the way they do? That is a puzzle that is confronting evolutionary biologists, and it's a problem that's attacked in all sorts of ways. Now, I'm only going to touch on just one way that biologists are addressing that issue by studying closely related groups of species that have a single common ancestor, that they've been derived from this ancestor relatively recently, and they diversified in various directions. These are called adaptive radiations, and there are some spectacular examples of them in nature. Hundreds of species of cichlid fish, for example, in the Great Lakes of Africa, and several lakes have several hundreds. In the Caribbean, we see Alleles lizards, 135 species of them, again, all derived from a single or a very small number of ancestral species in a relatively short space of time. Third example, in Hawaii, a group of birds that have diversified in the way that you see on the screen, about 50 species. They're called honeycreeper finches, and they too have been derived from a common ancestor. And there are plenty of other examples I could use to illustrate the phenomenon from the world of plants, and I have possibly jumped over it. All right, so I don't have the slide, but there are, in fact, very many examples in plants as well. So how do we address such a big question as the origin of such diversity? The key process that we would like to be able to understand is speciation. That is how two species are formed from one. But we need very much more information, and here's a brief summary of the multifaceted studies that biologists do. What is the time frame of the diversification? That we need to know, and how did the environment change during that time? What are the important ecological factors governing population genetics, population dynamics, and ecological niches? What are the traits that have been involved in the diversification? And finally, what are the genes underpinning the diversification at phenotypic, that means morphological, what we measure, what we see, variation? Our chosen group to address these questions is a group known as Darwin's finches, and they occur on the Galápagos Islands. 18 species altogether, and mitochondrial DNA dating tells us that they all evolved from an ancestor in the last approximately million years, maybe a little bit more. Looking at those pictures, you can see they're not as spectacular as those other organisms. There's very little color variation, for example. But where they do, where these species do differ is in body size and in beak size and shape. Now, beaks are instruments for picking up and dealing with food, so this is an ecological radiation, one in which ecological factors are important in the diversification process. They're young radiation, as I've said before. Many of the islands, the Galápagos Islands, are still in the pristine state, and the island archipelago, the archipelago of islands, is a very dynamic environment. It's volcanic in origin, lying astride the equator, and they owe their origin to a volcanic hotspot, which is underneath, I haven't got control of this, but anyway, below the western island of Fernandina, and the plate, Nazca plate, geophysical plate on which these volcanic islands sit, is moving towards the South American continent at the rate of 60 kilometers per million years. Thank you. And this is where, here's the hot spot, there's the direction of movement. 60 kilometers per million years. You wouldn't see it happen. Oceanographically, climatically, also over the period of the last million years, the islands have undergone a very big change. And that's due to cyclic oscillations in global temperature from full glacial to interglacial conditions about 10 times in the last 1 million years. And the principal effect, as far as we know, on the Galápagos has been the lifting and suppressing of the sea level. Now, this is what the Galápagos archipelago looks like now. We're in an interglacial period. But go backwards a bit in time when it was much colder, and the sea level was lower, with water being locked up in polar regions as ice. And going back even further, the islands even coalesced into larger land masses. So switching from glacial to interglacial conditions would have seen a complete reversal of what I've just shown as a sequence. So against this background, dynamic background of change in the archipelago, how do we explain how 18 species were derived within a space of 1 million years from one? I said at the beginning that speciation is a key process. And here I have an extremely simple diagram of how we conceive of this process. A lineage, a population, splits into two derived lineages, and they diverge to the point at which sometime in the future, they're two different populations, or rather members of the populations, are incapable of interbreeding and exchanging genes even if they attempt to do so. But the process is a very gradual one. Charles Darwin had a grasp of the bare essence of this process without knowing anything about genetics. His idea was that spatial separation of these lineages would facilitate the process of divergence. And I'm going to put his views into modern language with a cartoon here that represents our thinking of what is known as the classical model of speciation. And it is a three-step process, starting with the arrival in this new environment of colonists from the mainland. Everything is hypothetical. We don't know any of the details, in fact. But once the population was established on one of the islands, one can imagine the numbers building up and individuals dispersing to another island. And here we enter step 2, which is the colonization of a series of other islands and change in those other island environments. And why? Because of random processes, for one thing, but also for reasons of natural selection, adapting the populations to the particularities of each of the island environments. And I say that because the islands are not identical. Well, they're obviously not identical in size, but they're also not identical in the kinds of challenges that they pose to finches for living. So adaptation would have led to divergence, and then step 3 is the coming together of previously separated populations and further divergence, minimizing two things. One is interbreeding on the grounds that any offspring produced by two very different forms mating together would be relatively unfit. And secondly, a divergence due to the minimization of competition for vital resources like food. Now, that process of divergence is sometimes known as character displacement. And I'm now going to give you an example from our studies on this little island of Daphne that's too small on this diagram to be labeled. And this is what it looks like. It's an island of about 3/4 of a kilometer long, 120 meters high, and it's never been settled by humans, so it's in close to a pristine state. It is occupied by four species of finches in the genus Geospiza, ground finches. Now, I'm going to step back from the science for a moment and give you an idea of what it's like to work on the islands, and then step forward back into science. There's nothing on the island that we can live on, no water, no food. And we have to take everything, including all of our equipment, having put it into quarantine for 48 hours beforehand to make sure that we don't introduce anything that shouldn't be there. There's a crater in the middle of the island The birds go everywhere, and so do we. We have to follow them. They nest in the cactus bushes that you see on the flanks of this volcanic crater. We sleep in a tent. We cook in a cave that doubles up as a laboratory. And we, like other people who study bird populations, capture birds in nets, take out the birds, and before the sun gets really strong, take them to a cave, shady cave, where I weigh and measure the birds, and then put onto their legs 4 colored bands in total, so that we can, when we release them, recognize them. The colors are coded in unique ways. Each individual, when released, has a unique signature that we can observe through binoculars or even with the naked eye. But before releasing, I hand the bird over to Rosemary, who takes a small drop of blood by puncturing the wing vein here, producing a very small drop of blood, stores it in dry condition, we take it back to North America for DNA analysis. The focal species is Geospiza fortis, the medium ground finch. Now, by measuring, banding, and releasing, and then observing literally hundreds of these individuals, we know that those individuals who are seen feeding only on small seeds are very small in beak size. Those feeding on small, medium, and large seeds are very much bigger in beak size. So the species, as a population, is a generalist in terms of feeding with some degree of morphology-dependent specialization. There's another species on the island that I will only refer to briefly, the cactus finch, that is a specialist on cactus products. Now, the first species, being more of a generalist and varying more, is presumably genetically variable as well as phenotypically variable. And I say that because from an ancestor that we can infer, it must have undergone some change to become what it is today. And that implies some genetic variation in the past. So presumably there's genetic variation still in the population underlying the phenotypic beak size variation that we measure. And that turns out to be absolutely true by our measurements. We have measured the average of the father and the mother beak size, which is now on the horizontal axis, and the average of the sons and daughters, which is on the vertical axis. And so there's a very strong predictive relationship between offspring and their parents. The heritability, a measure of the inheritance on a scale of 0 to 1, is really very high, as indicated by the slope of those regression lines. Well, genetic variation is available then for natural selection to shift the population one way or the other. We didn't have to wait long before we observed a natural selection event doing exactly that. And it occurred as a result of a drought, which converted this normal view of the seed supply, lots of annual plants, perennial plants there with small clusters of seeds in the tops, converted that into a scene like this, where all of those plants died and the finches were dependent upon whatever seeds were on the ground left available to them. 80% of the fortis population, the medium ground finch population, died in this drought, and their average beak size underwent a strong shift upwards. The reason is simply told that during the early part of the drought, finches fed on small, easy-to-handle seeds, and when that supply was depleted, they had to turn alternatively to large and hard fruits, woody fruits, in which there are 5 seeds. They had to crack those open to get at their seed reward. Only the big beak birds could do that. They survived. The small beak birds had a high mortality rate, and so the average beak size went up until the rains resumed in 1978, and from then on there was no further selective mortality. So, natural selection occurred. Now, because the trait is heritable, then the next generation, the genetic factors that were inherited by the survivors should have been passed on to their offspring, and they indeed were. We measured the offspring and found that they were, as predicted, large like their parents. So evolution had occurred by natural selection on a heritably varying trait for biomechanical reasons that we could understand when the environment changed. The next thing we learned about natural selection occurred many years later, but it started with an enormous perturbation of the climate, an El Nino event of unprecedented in our history existence. Occurred in 1983 when 1.3 meters of rain fell on this little semi-arid island over a period of 8 months. And I'm going to show you what it did to the island. Here's a dry season picture of the island. The rains come in the early part of the year and convert the island into something that looks more green. In the foreground is this key plant, Tribulus, the plant whose woody fruits made the difference between survival and non-survival in 1977. Well, the rains came down and they caused a terrific amount of growth, principally of vines, and this plant was smothered by this enormous, dense array of herbs and vines that grew everywhere, up the trees, over the cactus bushes where the finches nested. We had difficulty finding the nests, and you could imagine, in clumps like this. And even in the next year, you could see the effects of the El Nino preceding with these vines draped across the trees. So the island was converted from an environment with small, medium, and large seeds into something dominated completely by small seeds, an overabundance of small seeds. No selection. Everybody survived very well. There was no fitness variation related to beaks. Everything was fine. But this was the key event, in our view, as we later discovered. This is a new species to the island, Magnerostris, the large ground finch, that previously had come in their ones and twos as vagrants and then gone back as soon as the rain came, presumably gone back to the island where they were hatched. But in 1983, 2 females and 3 males stayed to breed. And over the years, they built up a very large population until the time of the next drought in 2004, there were about 150 of these birds on the island. Now you can see with the figure at the bottom there what massive jaws this bird has and how easy it would be for it to crunch open a tribulus fruit, which is actually in its jaws, and extract the 5 seeds on the inside. So, under the conditions of diminishing food supply and the need to turn to tribulus in 2004, Magnerostris, top right, outcompeted Fortis on the left-hand side, and the large members of the Fortis population died to a disproportionate extent. The small members, bottom left, also were dying at a high rate, but not quite at the same high rate. And so the advantage went to them, and the difference between Magnerostris and Fortis over here, as a result of this differential survival, that difference increased. That's an example of character displacement that I mentioned earlier, and resulted in this drop in the average beak size in 2004. And there is the figure that I introduced the subject. So that led us into a question of underlying genetics. what are the genetic underpinnings of beak size that make such a difference in terms of survival or non-survival under conditions of stress? We knew there was additive genetic variation because we knew we had an evolutionary response to natural selection. But what were the key genetic factors that underpinned the development of beaks? For this, we had to, not being geneticists ourselves, we turned to a collaborator. Leif Andersson is our main person here, and the work that I'm going to be talking about was done principally by Sangeet Lavichani on the left-hand side. This is what they did. To look for genes associated with beak size, first of all, they sequenced all of the genome of all of the species of Darwin's finches and many individuals from different island populations, including Daphne Major. And then they set up comparisons of finches with different morphological properties, big versus medium beaks, medium versus small, and large versus small, in paired comparisons, and then looked along the genome to see where the paired species differed most of all. And they differed most of all in each three of these comparisons at this one locus here in purple, characterized by HMGA2, a known gene, a gene known in cats and rats, in mice and dogs, and in us humans, to vary in association with body size. Well, interestingly, in the finches, it varies principally with beak size, with our data showing one associated with large size, and we call it LL for obvious reasons, and one associated with small size. Even more interesting is that survival, fitness, in the drought of 2004, as indicated here for these three genotypes, was not only associated with beak size, but with genotype, the genetic characters that underpinned beak size. So the LLs had a survival of only 30%, whereas the small birds with an SS genotype survived more than twice as well. And the net result was that the allele, the L and S alleles at that locus underwent this extraordinary shift and stayed that way for the rest of the time. So we know quite a bit about this association between fitness and variation, and a little bit about genotypic variation as well. We then extended the work to 28 loci that were discovered in association with beak size. And I have 2 points to make about the discoveries that we made there. Firstly, as much as almost a half of the variation in beak size and beak shape could be accounted for by just 6 of genetic loci identified. That in itself is a very high number. Take out the effect of correlated body size, and the number is still very high, with HMGA2 being the principal one, and ALX1, another gene associated with beak shape, also having a very high contribution to make. My second point is that HMGA2 occurs also in body size as well as beak size and shape variation. Now, you remember there were 4 genes at that locus. HMGA2 was the one I mentioned, but that independent association with body size and beak size suggests to us that actually that locus is a supergene. That is, 4 genes are clustered together with different functions. In one case, at least one, it's body size, and in at least another case, it's beak size. So I will skip over this one and just summarize at this point and say, I've demonstrated that natural selection occurs when the environment changes. Other species determine the direction of the evolution, up or down in our case, and both of these are core ingredients of speciation. Now, I haven't said anything about interbreeding, and I'm happy to hand over to Rosemary at this point.
Oops. So Peter has told you about natural selection, but the evolution by natural selection requires genetic variation. Now, the amazing thing, when we first did all the measurements of Darwin's finches, we found that they were extraordinarily variable in each population, almost twice as variable as any bird that you normally see outside, like a song sparrow or something like this. So the question was, how is this genetic variation maintained on which selection acts? Because we knew that the Darwin's finch populations are small, they're isolated, they're subject to repeated droughts through El Nino, which occur about once every 10 years. So we'd expect them to become inbred, lose genetic variation, and eventually go extinct. But the answer is very rare hybridization. So what I'm going to do in this part of the talk is to look at the causes and the evolutionary consequences of this rare episodic introgression of genes between the species, and then tell you about one of the most exciting parts of our study, which is the formation of a new lineage, which we have followed from its inception all the way up through over 6 generations. So first of all, we have to know what are the pre-mating barriers between the species, and why does this barrier very occasionally leak? Well, we can't use plumage because all ground finches, not just the 4 on Daphne, but all ground finches have the same plumage. Males are black, females are brown, and streaked. They're also similar in courtship display and the types of nests. But they do differ markedly in size, they differ in beak shape, and they differ also in song. And its song, it turns out to be very important. We did experiments in the field with controls using museum mounts and playback of song. And this showed us that individuals really discriminate between their own and another species by appearance, morphology, appearance alone in the absence of song, and by song in the absence of appearance. And as I say, song turns out to be really important. And everything actually, now what I'm going to talk to you about, hinges upon this fact that they are what is known as closed learners. 50% of birds learn their song, around 50%. Others, like ducks and geese, don't learn their song. But these are closed learners, and the song in Darwin's finches is learned in a very short, sensitive period of time, between day 10 and day 40. And at this time, they're being fed by their parents, and all the time the male, the father, is singing. The females don't sing. But once this song is learnt, it is retained for life, and life can be as long as 17 years. As adults, they pair according to the species song they learnt early in life, and remain this way. So this pre-mating barrier is based on learning, and so as such it is vulnerable if the birds didn't learn another species' song. And this can be breached for 2 reasons. One, there can be nest takeover. So if on this side, on this side, the scandens, which is slightly bigger than Fortus, can take over the nest. Usually it turfs out all the eggs, but occasionally one is left behind. So that grows up as a Fortus in a scanden's nest and learns of scanden's song. It also happens with the death of the father, and as I said, the females do not sing, so the young in the nest learn the neighbor's song, and if that neighbor is another species, it will learn that song. And they learn such a pure rendering of that species' song that when they sing it, when they grow up and sing it, they are attracted by that species of the song that they're singing. So when the size difference is small, when the size difference is very large, then they sing that song, when they sing a Magna Rostris song, and a Magna Rostris would whip in and just just beat the living daylights out of it, and it doesn't survive. But when the species is small, the difference between species is small, then learning another species' song can lead to hybridization. And this happens about 1% of each breeding season. So Peter has shown you this, and we had these hybrids, which for the first 10 years of the study, we followed these 10 hybrids between Fortis and Scandens, hoping to see maybe some genetic incompatibility between them, but none of them survived to breed. And we didn't know whether this was because of genetic incompatibility or whether it was just these happened to be very dry seasons, and a lot of birds were dying anyway, a lot of young birds, and it could be that just they were dying. And this seemed to be the correct answer, because after this El Nino, when the island was converted from a hard seed producer to a small soft seed producer, and under these conditions of masses of soft seeds, then the hybrids began to survive. But we could still ask, how fit are the hybrids? Are they viable? Are they fertile? And is there any evidence of genetic incompatibility? So for the next 3 years, when a large amount of rain fell, we looked at the survival of the parental species, Fortis and Scandens, and here are the survival curves of Fortis and Scandens. And then when we looked at the hybrids that were produced, we found that, to our astonishment, the hybrids were surviving as well as, if not better, than the parental species. So there was no evidence of genetic incompatibility, and the hybrids backcrossed according to the song that they had learnt early in life. Now, another thing happened at this time. In Scandens, the cactus finch, it had a biased sex ratio. There were many more males than females produced. In fact, at one point, there were almost 100 males to 16 females. And so what happened then is that the Scandens males were desperate for females. They bred with the female hybrids, and but the male hybrids were outcompeted by the Scandens males for both females and for nest sites. So there were female hybrids were backcrossed into Scandens, so genes flowed mainly from the Fortis into Scandens. And so then what happened is that the Scandens' average beak size or beak shape became blunter and blunter and blunter and more Fortis-like. So they converged both genetically, the top graph, and morphologically by our measurements. Those Fortis and Scandens began to converge. But we wanted to know what were the specific alleles that contributed to these changes in beak shape. So we sent all our blood samples over to Leif Andersen's lab, and we had them both before and after this hybridization effect. And they found 2 really very important genetic alleles. One of them was ALX1, and this we knew from humans, because a mutation in ALX1 is largely responsible for cleft palate in humans. So it was obviously something to do with the cranial facial development. And it came in 2 forms, which we labeled ALX1B for blunt, because all the blunt-beaked birds, like Magnorostris, had this allele, whereas all the pointed-beaked birds had a different variant, which we called ALXP. And so Scandens had ALXP, and Magnorostris and Fortis ALXB. There was also another gene, RRIQ1, which binds with a calmodulin signaling molecule, which we'd already shown was with a collaborative work with people in Harvard. We'd already shown that that is actually present in the excuse me, in the developing embryo. So this was very exciting. We knew the genes that were involved. And so what we did by sending all our blood samples over before and afterwards, they were able to track ALX1 blunt going from fortis into scandens. So we were left with a huge amount of morphological variation that had increased in scandens. These were 3 birds that are scandens that we'd taken out of the same net in 2012. The top one has a very pointed beak, which is identical to scandens before hybridization, and the other 2 much blunter, which had ALX1B in them and were much more fortis-like. So we had published this work, and we said that increased genetic and phenotypic variation could fuel a rapid change along a different trajectory. And we never, ever thought that we would see this happening. But we did, and this was in the formation of the new lineage of finches. What happened was a finch arrived on the island. It was like a fortis, but like a blown-up fortis. Fortis is usually 17 grams. This one was 28.5 grams. It was a young bird, and when it started to sing, it had a completely unique song, which we had never heard on the island before. It took ages to breed. It was a big bird. It managed to get a superb territory, and it was not until 2 years. But first, we wondered where it came from, and the genetic data showed that it was a conirostris that had come all the way from Hispaniola and landed on Daphne. So as I said, it took ages to breed, all the wrong appearance, all the wrong song, but eventually it bred with a fortis who had lost its mate and paired up with him in this beautiful territory, produced a large number of offspring. All the offspring had genetic transmission from 5110, the bird that arrived. All males sang 5110's song. And then along came the 2.5-year drought that Peter referred to, and all these birds went out except these 2, an inbred brother and sister. When the rains came back, the brother and sister bred with each other, produced a large number of offspring. All but 9 of them survived. We had a daughter breeding with her father, a son breeding with his mother, and the rest of the sibs breeding with each other. All the birds showed genetic transmission from 5110. All males sang the 5110 songs, and all were large like 5110. But interestingly, there was an allometric shift. So in red is the conirostris from Hispaniola, where the bird came from, and this is 5110, the black square. And in green is the fortis population. In blue are the big bird population. And you can see that actually there's been an allometric shift, and that they had a large bill relative to their body size. And they loved these. Oh, and I should say the reason for this is that these 2 genetic factors, HMGA2 and ALX1, are very closely linked on the same chromosome, but there's a recombination hotspot between them. So you can get a shift between size and beak shape due to this recombination, so you couldn't get all combinations. And the one that came out with what we call the big bird group is the one that had a relatively large beak for its body size. Now, it loved tribulus seeds, and we were wondering how efficient it was at cracking tribulus seeds compared with Magnorostris, and we found that it was just as efficient. But if there was another drought, having a big beak and a small body, it would require less food, so it could even outcompete Magnorostris for eating the tribulus seeds. So the new lineage is behaving like a separate species. It's much larger than Fortis, its nearest relative. It differs in size. If we look at beak length over beak depth, we can see Fortis in blue, Magnerostris in green, where Magnerostris outcompeted the largest Fortis. It left a gap, and the big bird moved right in. It sings a different song from all other species on Daphne. The songs on the left are 3 generations of big bird song, very different from Fortis song here at the top, Scandin's song here, and Magnerostris song there. It holds territories in the same part of the island. They're contiguous territories with each other, and they overlap with Fortis, Scandens, and Magnerostris, who completely ignore them. Thus, the new lineage is functioning as a separate species. Will it die out through inbreeding depression? Well, it might. It certainly is getting more inbred, but it started with a huge amount of genetic variation, and really it's no more inbred than many of the other populations on the islands. Will the genetic variation be augmented through gene exchange? It might. It goes around a lot with Magnerostris, and it could even there could even be gene exchange between it and Magnerostris, but so far not yet. Whether it survives or not, this new lineage gives us insight into how a new species can arise and either persist or go extinct. So in summary, when we first started, there were only 2 species on the island, Fortis and Scandens. Hybridization after this extraordinary El Nino event brought them together. The Magnerostris came in about this time, outcompeted the large Fortis, and the big birds moved in. So a very different scene after 40 years. So how important is introgression as a source of variation for adaptations to new environments? I should just say that the very first time we sent a letter, sent a manuscript in about this, it came right back with the words, didn't even go out for review, birds don't hybridize. And of course, now we know, and that was thought to be so then. Hybridization was thought to be hardly happening at all. Of course, we now know that it's happening everywhere. It's happening in viruses, as we know with the H5N1 strain that has just arisen, bacteria, plants, and in all groups of animals. It also happens in our own ancestors, as Finches has shown. Source of variation used in adaptation in new environments have come from Denisovans into Neanderthals to our own ancestors. So we thank our many graduate students, postdoctoral fellows, collaborators, and our daughters who are with us on the island for much of the time. But I want to just leave you with one message. I love this Nassau image of our planet because it captures both the uniqueness and the fragility of our planet. We have shown you, with biodiversity being its distinctive feature, we've shown you that environments and populations are dynamic, constantly changing, and for a healthy environment, we must keep them both capable of further natural change. Thank you.